Welcome. Malignancy criteria or hallmarks of cancer cell. Team criteria of the cancer cell. We can call it cellular hallmarks distinguish a cancer cell or criteria from a normal human eukaryotic cell. Cancer cell fit or surviving and reproducing. The cancer cell competing, surviving and reproducing. A list of terms we will use in these lectures. A series of lectures about the criteria of the cancer cell on cellular level. Apoptosis. What is apoptosis? Apoptosis is a form of programmed cellular death. When the cell feel that there is something in its mitosis, it commits suicide or push itself to the death. Mitosis is a form of cellular division that results in two daughter cells. Mitosis, a process in which the cell divides and the end result is two daughter cells. Telomere, these are the sequence located at the end of the chromosome. These have specific sequence of nucleotide, and these sequence shorten at the end of each mitotic cell cycle. The telomere is the end of chromosome. Angiogenesis is the process of developing new blood vessel from pre-existing one, and finally metastasis. Metastasis, this is a process by which a cancer cell or cancer itself spread from its origin. Process by which cancer itself spread from its origin. The eukaryotic cell is the smallest functional unit that can produce daughter cells in the human body. It's a word that comes from meaning small in Latin. Cell means small. It's able to produce two daughter cells through a process called mitosis. This process is tightly controlled. Cells are present throughout our human bodies that make up tissues, and these tissues make up organs, and so cellular homeostasis is very important in tissue and organs. These cells, as you can see, made up of number of organelles. The cell form the organs, and the organs form the system of our body. Here inside the cell there is organelles, the nucleus, the nucleolus, as well as mitochondria, and mitochondria found in the cytoplasm, mitochondria, and the lysosomes, the centrosomes, other organelles located in the cytoplasm. The production of cells progeny is tightly controlled in the machines called the cell cycle. Cell cycle. The cell cycle is a series of events that take place in cell, ending to its division and duplication of its DNA, known as DNA replication, to produce two daughter cells. In the nucleus of the cell, the cell cycle is divided into three period interphase: the mitotic phase, and shown by M here in the cartoon, M, and cytokinesis, which is shown of the two daughter cells here, two daughter cells, the end result. During the mitotic phase, again the cell split into two distinct daughter cells. the new cell is completely divided. In this cell cycle, there are checkpoints. To ensure proper division of the cell, there are several control mechanisms that occur during the cell cycle. There are known as the cycle checkpoints. These are located gap phases. The G phase is shown in this cartoon. G phases is shown. This is germinate to what distinguishes a cancer cell from that normal cell, and lead us to the characteristic number 1, or the hallmark number 1, replicative immortality. The normal human cell have finite ability to undergo mitosis due to what is known as the end replication problem. This is largely due to its part, the telomeres of the chromosome, which are the ends of the chromosome that get shorter after each mitotic division. This the shortest sequence of the DNA get shorter every division. One of the first people to observe this, Dr. Hayflick, this person, Hayflick, and when cells stop dividing, they are known to have reached Hayflick limit. And once cells reach Hayflick limit, they can no longer divide. Dr. Hayflick Leonard Hayflick discovered that the cultured normal human cell have limited capacity to divide, after which they become senescent, a phenomenon now known as the Hayflick limit. About 60 division or less, and that is the limit of the normal cell division. A cancer cell from that of normal cell lead us to a cellular hallmark of cancer. Number 1 is the replicative immortality. Normal human cell have finite ability to undergo mitosis, because what's known as the end replication problem. This due to telomeres of the chromosome, as we mentioned, which are the end of the chromosome that get shorter after each mitotic division. Dr. Hayflick, when cells stop dividing, they are known to have reached Hayflick limit, and once cells reach Hayflick limit, they can no longer divide. Normal human cell can only divide 40 to 60 times before they can't divide any further. Later, the researcher found that the cause of Hayflick limit is the shortening of the telomeres, or portions of the DNA at the end of the chromosome that slowly degrade as cells replicate. Hayflick used his research on normal embryonic cells to develop a vaccine for polio, and from Hayflick published directions, scientists developed vaccine for rubella, rabies, adenovirus, measles, chickenpox, and shingles. And they go in G0 phase of the cell cycle, called cellular senescence. You can appreciate the ends of the chromosome, those are the telomeres. The normal cell can stop dividing once the telomere become too shorter. Cancer cell is able to greatly exceed what is known as Hayflick limit and continue to undergo mitosis. Cancer cell can greatly exceed Hayflick limit by using an enzyme known as telomerase. Telomerase is able to elongate. What is telomerase? Telomerase enzyme responsible for maintenance of the length of the telomeres by adding of guanine rich repetitive sequence. Telomerase activity is exhibited in gametes and stems and tumor cells. Telomerase, the cancer cell can greatly exceed half-life limit by using an enzyme known as telomerase. Telomerase is able to elongate after they get too shorter to sustain proliferation. Telomerase is seen to be expressed at high level in several cancer types. This is a cartoon for telomerase. It's an enzyme that's reverse transcriptase enzyme that adds short the hallmark number 1, as we mentioned, repetitive immortality by cancer cell, the cancer cell being immortal. By cancer cell being immortal, they are able to continuously proliferate, and also they can pass on genes to daughter cells that are mutated. This leads us to hallmark number 2, known as genome instability. Gene was a unit of inheritance that consisted of 2 alleles. In this hallmark, we'll see how genome instability gives rise to tumor genesis. The characteristic number 1, or the hallmark number 2, genome instability. In normal eukaryotic cell, they bear 22 autosome and 1 repair of 6 chromosome, reside in the nucleus. Normal cells undergoing DNA synthesis during the cell cycle. It detects a mutation which occur in gap phases of the cell cycle. The cell is able to stop the cell cycle, repair the mutation, and then re-enter the cell cycle. Genes that are involved with stopping the cell cycle from mutation is detected are known as tumor suppressor gene. So think of these genes as being the brake in your automobile. If you see traffic coming, you put on the brake to stop your car or your automobile. Tumor suppressor gene works similarly. However, cancer cells differ from the normal cell due to their ability to bear an abnormal amount of chromosome within the nucleus and also still be able to undergo mitosis. Genes that are mutated in cancer or lost in the cancer contribute to genome instability. These are your brakes, known as tumor suppressor gene. Conversely, there are genes that become overexpressed or mutationally activated, and these are known as oncogenes. Genomic instability is a fundamental characteristic of essentially all cancer. It refers to the increased rate of alteration in the cell genetic material, including mutation, chromosomal aberration, epigenetic modification. These alterations disrupt normal cellular processes and contribute to development and the progression of cancer. What is the causes of genomic instability? Defects in DNA repair mechanism. Cell possesses intricate DNA repair pathways to fix error and damage that occur during DNA replication or due to environmental factor. Mutation in gene encoding DNA repair protein can compromise these pathways, leading to build-up of mutations. Telomere dysfunction. Telomeres are protective caps on the chromosome that shorten with each cell division. When telomeres become critically short, cell can sense DNA damage and trigger cell cycle arrest or senescence, permanent cell growth arrest. In cancer cells, telomere dysfunction can lead to chromosome fusion and other aberrations. Oncogene activation. Certain oncogene genes that promote cancer development can directly or indirectly contribute to genomic instability. For instance, some oncogenes involved in cell cycle regulation can bypass checkpoint, allowing damaged DNA to be replicated. Chronic inflammation. Chronic inflammation creates a stressful environment for cells, leading to production of reactive oxygen It can damage DNA. This persistent DNA damage can overwhelm repair mechanisms and contribute to genomic instability. So the causes of the genomic instability: defect or in repair, telomere dysfunction, oncogene activation, chronic inflammation. Types of genomic instability: chromosomal instability. This the most common type, characteristic by gains or loss of entire chromosome or large chromosomal segment. Interchromosomal instability. This refers to mutation and rearrangement within chromosome. Microsatellite instability. This involve alteration in short repetitive sequence called microsatellite. Epigenetic instability. This encompasses change in gene expression pattern that don't involve alteration in DNA sequence itself. These changes can be caused by modification to DNA methylation pattern or histone tail structure, which can lead to abnormal gene activation or silencing. Genome instability promote cancer development by inactivating tumor suppressor gene, activation of oncogene, increasing tumor heterogeneity. Targeting genomic instability in cancer therapy. Understanding the mechanism of genomic instability hold promise for novel cancer therapies. Strategies include inhibitors, exploit defect in DNA repair pathways, targeting telomerase, the enzyme that maintain telomere, inhibit telomerase activity, can induce senescence in cancer cells. Immunotherapy. Tumors with high mutational burden can express unique protein antigens that trigger an immune response, and this is a promise for new therapy. Cell activities, which immune system's ability recognize and attack these cancer cells, improve the immune system ability to attack these tumor cells. The G1 phase of the cell cycle as a vital checkpoint wherein anti-growth signal exert their influence to block cellular proliferation. Genomic instability. Most cancer cells are able to circumvent normal growth suppressor signal in G1 checkpoint in order to continue to proliferate. How cancer cells are able to evade growth suppressor signal? One mechanism is the tumor suppressor gene called retinoblastoma, RP, actively inhibits cell passage through the restriction point in G1 cell. Cancer cell with mutated RP remove this gatekeeper or ongoing cellular proliferation. Cell activities at the cell cycle level, particularly G1 phase, the cell cycle as a vital checkpoint wherein anti-growth signal exert their influence to block cellular proliferation. Hallmark number 2, genomic instability. Most cancer cells are able to circumvent normal growth suppressor signal in G1 checkpoint in order to continue to proliferate. Again, how cancer cells are able to evade growth suppressor signal? One mechanism is the tumor suppressor gene called retinoblastoma, inhibits cell passage through the restriction point in the G1 cell cycle. Cancer cell with mutated RB remove this gatekeeper or ongoing cellular proliferation. Another tumor suppressor gene called B53 is lost in many cancer types. This allows for the cell cycle to progress despite DNA damage and other cellular stressor that would kill a normal cell. With this gatekeeper, genes are lost or mutated. Cancer cells are able to sustain proliferation and resist cellular death. Normal cell can initiate cell death, also known as apoptosis, in response to an abundance of DNA damage, DNA mutation, as well as other cellular stress from external factor. As we stated in a previous slide, cancer cell can continue to cycle through the cell cycle while repairing such DNA damage in other cellular stresses. Cancer cells are also able to resist cell death by upregulating pro-survival protein to avoid cell death in the presence of these stress. One example of the pro-survival protein that is seen overexpressed in many cancer types is the pro-survival protein BC1, BCL1, anti-apoptotic family members such as BCL2, BCL, XL, MCL1, CED9, A1, BFL1, and also pro-apoptotic family members such as BAX, BAC1XS. In many cancer types, we see BACL, BC1XL, BC12 family member overexpressed, and that is shown here in the method used in molecular biology called immunohistochemistry. This BC1 and XL overexpressed in lymphoma, and you can appreciate the brown B positive cell in this immunochemistry slide because cancer cells are able to resist cell death. They are able to sustain proliferation within normal cell. Growth factor signaling is also tightly controlled for tissue homeostasis and other cellular function. Cancer cells are very different and they have the ability to proliferate, as I mentioned in hallmark 1. Loss of too much suppressor gene as well as overexpression of oncogenes such as RES and SARS, SERK, and many other cancer cells also have the ability to simulate the normal cell that surround them in what's known as tumor microenvironment to provide them with essential growth factor. One growth factor that's well studied cancer researcher is called the epidermal growth factor. Epidermal growth factor can bind to the receptor which is expressed on cancer cell known EGFR to activate an oncogene well studied in cancer research called RES, have a plethora of downstream effect that positively regulates cell proliferation of cancer cells. So, quick review over hallmark from number 3 to 5. Normal cell when it detects DNA damage, P53, a tumor suppressor gene, stop the cell from cycling, put the cell into cellular arrest, repair the DNA, and the cell is able to go back into the cell cycle. That is one option. If there is too much DNA damage, the cell is signaled to go into apoptosis, and then the dead cell is then cleared by phagocytes, and this leads to cellular and genetic and tissue homeostasis. In cancer cell, there is a loss of mutation of B53 where it doesn't function correctly, and when the cell has apparent mutation, the cell is not able to leave the cell cycle, and then instead it continues to proliferate and go through the cell cycle unchecked, doesn't undergo apoptosis, and you get genomic instability instead. So here is the first five hallmark or the 5 characteristics we have covered. Number 1, replicative immortality, genome instability, evading growth suppression, sustained proliferation, and resist cellular death. Number 1, repetitive immortality, genome instability. Number 2, genome instability. Number 3, evading growth suppression. Number 4, sustained proliferation. Number 5, resist cellular death. This is all for this lecture. It's somewhat long, but you can review these points in this print out. I leave it in your resources, and you write down some notes. Any question? Thank you, and see you in the next leakage.